faculty exchange program, we are met here between both the prestigious institutions, one from Government Degree College for Women, Jagitial, the another prestigious institution in Twin Cities, that is RBVRR Women's College, Autonomous, Narayanguda, Hyderabad. So this faculty exchange program leads to enhance the knowledge between these two institutions and benefit. So for the welfare of the benefit of the students, the exchange of the faculty means, so these two colleges have then very eminent and experienced faculty. These faculty is going to exchange their views, knowledge, and experience and skills regarding of our concerned subject. So that's why it is very, very useful program. I hope you clearly understood what about I am sharing my knowledge among you. So yesterday we are discussed gene expression. In this gene expression, we are clearly explained about the replication of the DNA and the transcription process. Also, we are discussed yesterday. So remaining topic we will continue today. So yesterday we are discussed about the transcription is divided into 3 stages. That is this slide is visible. Anybody?
Yes, sir.
Okay, so initiation, elongation, termination. These are the 3 steps we are already discussed yesterday. So after this transcription or termination process, so post-transcriptional modifications is also there. In this post-transcriptional modification, it is a process in which precursor messenger RNA is converted into the matured messenger RNA, that is mRNA. So mRNA is a messenger RNA. It is a matured messenger RNA. It is before the formation or converted from precursor messenger RNA. It is formed after the transcription process. So this post-transcriptional process or modification has 3 steps or 3 stages. One is a 5' capping, or the second one is the 3' polyadenylation, and third one is RNA splicing. So first one, 5' capping. That is a 5' capping is the addition of 7-methyl guanosine cap to 5' end is the first step in post-mRNA processing. So this step occurs co-transcriptionally after the growing RNA strand has reached 30 nucleotides. Whenever this RNA strand has reached 30 nucleotides, then automatically this 5' capping we are observed 7-methyl guanosine cap at the 5' end of the on the mRNA. Then 3' polyadenylation. So polyadenylation means many. So adenine is a many number of the adenine is attached to the tail. So that we can call as a polyadenosine chain. That is a poly-A tail. So this is the second step. It is the cleavage of the 3' end of the primary transcript, following by addition of polyadenosine A tail or poly-A tail. So this is the second post-transcriptional stage. Then the last one of the post-transcriptional changes or modification is RNA splicing. So splicing is taken by this. This process is taken by spliceosomes. So this RNA splicing is the process by which introns are removed from the mRNA, and the remaining exons connected to form a single continuous molecule. The splicing reaction is catalyzed by a large protein complex called the spliceosome. Okay, spliceosome. So these are 3 post-transcriptional modifications we are observed in the entire all living organism. Then after transcription, the most important and multi-complexed stage of the sequential events of the gene expression, that is the translation. So this is the third sequential step of the gene expression, that is the translation. Okay, am I clear? This is gene expression, the gene expression, We know the previous class, we have discussed about the replication of the DNA and the transcription, which means from DNA to mRNA synthesis. Then the third multi-step process, that is the translation, the gene expression. In the third and last one, that is the translation. So in this translation, it is the process by which proteins are synthesized. So from mRNA to protein synthesis process, we can call as translation. Translation is a complex cellular process where mRNA molecules, including ribosomes, tRNA molecules, amino acids, amino acyl synthetases, which are enzymes, and energy sources like ATP, adenosine triphosphate, and guanosine triphosphate, GTP, and a number of factors act together in a highly coordinated way. Translation is a highly coordinated way. So these are all involved in this translation process. Okay, so the mRNA carries genetic information encoded as a ribonucleotide sequence from the chromosome to the ribosome. Then the ribonucleotides are read by translation machinery in a sequence of nucleotide triplets called codons. We know that codons, our genetic code is the triplet codons, So nucleotide triplets are called codons. Each of these triplet codes for a specific amino acid, the ribosome and tRNA molecules translate this code to produce proteins. Okay, we know that. So nucleotide triplets are called codons. So each code, the triplet codes for specific amino acid. For example, AUG is a triplet code. AUG codes for methionine amino acid. So that's why these triplet codes for a specific amino acid. The ribosome and tRNA molecules translate this code to produce proteins. Okay, translate code to produce proteins. These tRNAs have a site for amino acid. What is the tRNA? We know that transfer RNA. tRNA means transfer RNA. So this translation process, tRNA is most important RNA among different RNAs like mRNA, rRNA, tRNA, and snRNA. So different RNAs are small nuclear RNA is also there, but tRNA is most important in this entire translation process along with mRNA. So tRNA have a site for amino acid attachment and a site called an anticodon. Which codon? Anticodon. Remember, my dear friends, codon which is present on mRNA, at the same time anticodon site is present or situated or possessed on tRNA only. The site for a site for amino acid attachment on a site called anticodon. So anticodon site we are observing tRNA only, anticodon loop or anticodon which is present in tRNA only. This anticodon is an RNA triplet complementary to the codons of mRNA. So mRNA consists codons, whereas anticodon consists tRNA. Then amino acid tRNA synthetase are catalyzed. This is enzyme, my dear friends. Amino acid tRNA synthetase is an enzyme. This catalyzes the bonding between specific tRNA and the amino acids that their anticodon sequences call for. The product of this reaction is an amino acyl tRNA molecule. This is nothing but charged tRNA. So this is structure. This slide shows about the structure of this ribosome. So ribosome possesses a larger subunit along with the smaller subunit, my dear friends. Larger and smaller subunits. We know that in prokaryotic living organisms, 70th type of ribosomes are present, whereas eukaryotic living organisms possess 80th type of ribosomes. Along with these prokaryotes, whereas eukaryotes, these ribosome possess larger and smaller subunits. 70th type of ribosomes possess in prokaryotes, whereas 80th type of ribosomes are present in eukaryotic cells. My dear friends, each ribosome consists two subunits. One is the larger one, then another one is smaller subunit. Larger subunit of the prokaryote, that is 50S, and smaller subunit is 30s. But we are going to discuss here larger subunit and smaller subunit of the eukaryotic living organism or eukaryotic cell. That is, the larger subunit is 60s subunit and the smaller subunit 40s subunit. Am I clear? Am I clear? Larger subunit and smaller subunit. Then, so this in this translation is also process initiation, elongation, and termination. These are the three different stages we are observed in this translation also. So DNA replication is also 3 stages, whereas the transcription is also 3 stages, including translation is also we are observed are divided into 3 stages: initiation, elongation, and termination. In this initiation of a translation is divided into another. This initiation is also divided into 4 stages. The first stages of this initiation is dissociation of ribosome. What is the meaning of dissociation? Actually, ribosome is associated with larger and smaller subunit. So both they are going to point with the between this ion is magnesium ion. Magnesium ion is going to point between these 2 subunits, larger subunit and smaller subunit of the ribosome. So eukaryotic ribosome, eukaryotic living organism or eukaryotic cell possesses 80s type of ribosome. S means, you know that, S means Svedberg unit, you know. So this 80s ribosome into smaller subunit, that is 40s, and larger subunit, that is 60s subunit. Even though in this dissociation of ribosomes, the initiation factor 3 and initiation factor 1A binds to the 40s subunit, that is the smaller subunit, and prevents its reassociation with 60s subunit. So here what we are observed, my dear friends, initiation factor, these are the 3 factors are going to bind on 40s subunits. Why? Because this 60s subunit is going to reassociate with 40s. Then only this IF3 means initiation factor 3 and IF1A, that is initiation factor 1A, binds 40s subunit. So this is the prevent the reassociation of the smaller subunit and the larger subunit. Okay, then formation of 43s pre-initiation complex. So the second step of this initiation is formation of 43s pre-initiation complex. The first amino acid tRNA, that is F-met tRNA, binds to the 40s ribosomal subunit and forms pre-initiation complex. This initiation factor IF3 and IF1A stabilize this complex. What is the complex, my dear friends? Formation of 43s pre-initiation complex. So this 43s pre-initiation complex is going to stabilize it by initiation factor 3 and initiation factor 1A. Okay, so then the third step of the initiation stage of the translation, that is the formation of 48s initiation complex. So first one is the dissociation of the ribosome. 80s ribosome is going to dissociate. That is the 40s and 60s dissociate, and then formation of the 40s, 43s pre-initiation complex. Then the third one is the third step of the initiation stage is formation of 48s initiation complex. This mRNA joins to the 43s pre-initiation. See here, this is the 43s pre-initiation complex and forms the 48s initiation complex. This step requires energy from ATP, adenosine triphosphates. Then ribosomal initiation complex scans the mRNA for the identification of the appropriate initiation codon, and its identification is facilitated by specific sequence of nucleotide surrounding it is called COJA consensus sequences. So these COJA consensus sequences are present or referred, or these are present in only eukaryotic living organs. Whereas in prokaryotes, the recognition sequence of initiation codon is referred to as Shine-Dalgarno sequence. So COJA consensus sequence which are present in eukaryotes, whereas the prokaryotes, the recognition sequence of initiation codon is referred to as Shine-Dalgarno sequence. So here, two sequences we are observed in this slide. The first one is Kozak consensus sequences, which are facilitated to specific sequence nucleotide surrounding. Okay, then the prokaryote, where is the prokaryotic, sequences that is we can call as Shine-Dalgarno sequences. First one is Kozak consensus sequence, and the second one, that is the prokaryotes, we are observed Shine-Dalgarno sequence. Then the fourth subphase or fourth phase of the initiation stage, that is the formation of ITS initiation complex. So what we are observed in this four steps, first one is the dissociation of ribosome. The second one is the formation of 43 pre-initiation complex, and the third step is formation of the ITS initiation complex. 43S pre-initiation complex, and the third one is 48S initiation complex, and the fourth stage of the initiation stage, that is the formation of ITS initiation complex. So this initiation ends at the large 60S large subunit, that is the 60S ribosomal subunit joins the 48S initiation complex, causing the dissociation of initiation factor. The binding involves the hydrolysis of GTP, guanosine triphosphate. The step is facilitated by the involvement of initiation factor 5. So the first step, two factors we are observed, my dear friends, that is the IF3 and IF1A. The fourth step is needed or facilitated by this fourth step, that is IF5, initiation factor 5. So here three factors we remember, my dear friends: IF3 and IF1A and IF5. These are the three factors which are involved in the initiation step. Okay, then the second step of this translation, that is the elongation. So elongation of the polypeptide chain involves addition of amino acids to the carboxyl end of the growing chain. During elongation, the ribosome moves from 5' end to 3' end of the mRNA that is being translated. My dear friends, remember here in previous class, 5' end to the elongation process should be from 5' end to 3' end. This is very, very important, and remember, my dear friends. Okay, so the mRNA that is being translated. So elongation is also further divided into 3 steps. Initiation we are divided into 4 steps. As like that, elongation is also further divided into 3 steps. The first step of this elongation is binding of amino acyl tRNA to A site. So what is A site? Remember, my dear friends, actually ribosome consists 4 sites. How many sites possess in ribosome? 4 sites. Okay, so the first one, so one for mRNA, first site for mRNA, remaining 3 sites for tRNA. Ribosome consists how many sites? 4 sites. Okay, 4 binding sites. Sites means binding sites. So these 4 binding sites are located on ribosome. First for one binding site for mRNA, remaining 3 binding sites for tRNA. These tRNA sites are labeled as A site, P site, and E site. A site we can call as amino acyl site. A site we can also call amino acyl site. P site means here, P also here. P site means peptidyl site. So what is E site, sir? What is E site means after the elongation, termination, that is the exit site. So these are the 4 binding sites we are observed on the ribosome. Okay, so the ribosome 4 binding sites consist 1 for mRNA, 3 for tRNA. This tRNA labeled as A site, P site, and E site. Remember, A site, P site, and E site. A site So this elongation process, the first step is binding of amino acyl tRNA to A site. So amino acyl tRNA binds which site? A site. Okay, A site is the first site. Remember, my dear friends, A site is the first binding site on tRNA. Then what about P site, sir? P site is the second binding site of the tRNA. Here, the ITS initiation complex contains methionine. MET means methionine. It is the first amino acid, my dear friends. So this tRNA we can call as MET tRNA. So MET tRNA on the P site and the A site is free. So whenever this MET tRNA on P site, then automatically A site is free. Okay. Then the another amino acid tRNA recognizes the codon on A site and bind to it. The binding facilitated by elongation factor 1 alpha. 1 alpha. So this binding facilitated by which factor? Which elongation factor? 1 alpha. Elongation factor 1 alpha and requires energy from guanosine triphosphate, GTP. Is it clear? Okay, very good. Then the second step of this elongation process, that is the formation of peptide bond. Formation of peptide bond. Now the P site contains the beginning of the peptide chain of the protein to be encoded, and the A site has the next amino acid to be added. The growing polypeptide connected to the tRNA in the P site is detached from the tRNA in the P site, and a peptide bond is formed between the last amino acids of the polypeptide and the amino acid still attached to the tRNA in the A site. Remember, first amino acid is going to attach at A site, then only it reaches the P site. It is a continuously going on process. Then translocation. What do you understand about the translocation? Now, the A site has newly formed peptide while the P site has an unloaded tRNA. Unloaded means without amino acids. tRNA with no amino acids. That we can call as unloaded tRNA. Then the ribosome moves 3 nucleotide towards the 3' end of mRNA. 3' end of mRNA. Since tRNA like linked to mRNA by codon anticodon base pairing, tRNAs move relative to the ribosome, taking the nascent polypeptide from the A site to the P site and moving the uncharged tRNA to the E site. E means exit site. This process is catalyzed by elongation factor 2. My dear friends, remember EF2 means elongation factor 2. Okay, so in previous slide, what we observed, so this elongation factor 1 alpha required or facilitated for the binding process and requires guanosine triphosphate. Okay, at the same time, this entire process is needed that elongation factor 2, that is EF2. And the third step of this translation process, that is the termination. So termination is the last step, last stage. It is the end step of the translation. This translation occurs when one of the 3 termination codons move on the A site. Whenever these 3 codons moves on the A site, these codons are going to recognize by proteins called RF1. That is, namely, we can call it the released factors or RF1 factors. Released factor 1 or RF1. So this RF1 recognizes the UAA and UAG. These are the stop codons, Also, we can call as nonsense codons. At the same time, these termination codons of UAA and UGA is also recognized by RF2. So RF1 is going to recognize this of UAA and UAG. At the same time, RF2 is going to recognize on the A site of the ribosome, that is UAA and UGA. These are also stop codons. These are UAA, UAG, and it is recognized by RF1. UAA and UGA is recognized by RF2, release factor 2. It is also we can call as RF2 and RF1. So these two are released when these factors recognize for the stop codons, UAA or UAG and UAA and UGA. Then these factors trigger the hydrolysis of the ester bonds in peptidyl tRNA and the release of the newly synthesized protein from the ribosome. Okay, whenever the these factors RF1 and RF2 is triggered the hydrolysis of the ester bonds. Okay, ester bonds in peptidyl tRNA and then release of the newly synthesized protein from the ribosome. At the same time, the ribosome is dissociated from the mRNA and recycled and used to synthesize another protein. It is a continuation process. Then protein folding. So what we are going to understand about the protein folding, we know that protein structure in biochemistry of the proteins. So protein structurally we know that primary structure and secondary, also secondary structure, tertiary structure and quaternary structures. We are already studied about the structures of the protein. So protein, primary structure, secondary structure, tertiary structures and quaternary structures. Among these four types of the proteins, which one is very suitable and which one is very active for the biosynthesis process or the physiological process. So this protein folding is a process by which a protein assumes its characteristic functional shape of tertiary structure, also known as the native site. So primary, secondary, tertiary and quaternary, among these tertiary structure, also known as the native site, native state, it is functional shape. Then all proteins molecules are linear heteropolymers composed of amino acids. This sequence is known as the primary structure of the protein. So primary structure of the protein, we are observed linear heteropolymorphic composed amino acids observed in this protein structure. Then most of the proteins can carry out their biological functions only when folding has been completed, because three-dimensional shape of the proteins in the native state is critical to their function. The process of folding often begins co-translationally, so that the N-terminus of the protein begins to fold while the C-terminal portions of the protein is still being synthesized by the ribosome. Specialized in this, my dear friends, remember, the specialized proteins are called chaperones. So this is a competitive question, my dear friends. Remember, chaperones are the specialized proteins aid in the folding of the proteins. Chaperones. Okay, chaperones are the specialized proteins. Okay, chaperones are nothing but specialized proteins. Then after translation, post-translational modifications also we are going to discuss here. So many proteins synthesized by translation are not functional as such. Many changes takes place in the protein after synthesis, which converts into active protein. So these are known as post-transcriptional modification or post-translational modifications. Okay, then trimming by proteolytic degradation. Many proteins are synthesized as precursors, not the matured one, my dear friends. Then precursors, which are bigger in size than functional proteins. So precursor proteins are not functional. Whenever the complete structure of the protein, which is functional, but compared to the functional proteins, these precursor proteins are bigger size. Some portions of a precursor is removed by proteolysis. Lysis means breakdown. Proteo means the protein. In the protein structure, some of the unwanted or the some of the parts is removed by proteolysis is to liberate active protein. This process is called trimming. This process we can call as trimming. Okay, example of formation of insulin from proinsulin. Pro means precursor of the proinsulin means precursor of the insulin. Here the precursor, the proteins are synthesized on precursor which are bigger in size than functional proteins. Like that, pre-insulin. So the proinsulin is the primitive one which are precursor of the formation of the actual functional insulin. Actual insulin. Okay, insulin is nothing but protein, my dear friends. Remember, insulin is also a protein. So then intense splicing. So first one is the trimming by the proteolytic degradation, and the second one is intense splicing. Okay, so intense are intervening sequences in proteins. So these are comparable to introns. Comparable to introns in mRNA, inteins have to be removed and extends ligated in the appropriate order for the protein to become active portion or active protein. Okay, this is about intein splicing. Then covalent modification. Then covalent modifications. So these covalent modifications, proteins synthesized by translation are subjected to many covalent changes. By these changes, the proteins are converted to active or inactive form. The covalent changes include many modifications such as phosphorylation, hydroxylation, glycosylation, methylation, and acetylation process. So these are the very important changes, that is the covalent modifications. So this is about the translation, the entire translations of gene expression. If you have any doubt, please ask me, I will clarify. Any doubts? Nobody is respond.
No, sir.
Any doubts?
No,
my dear
sir.
friends? No doubts. Okay, I hope translation is completed. I hope you clearly understood about the translation. Translation is also three steps. First one is initiation, the second one is elongation, and third one is the termination. Okay.
I look like it.
Madam?
Yes, sir. Yes, sir.
Okay. So these are the students have so many doubts. Please you clarify.
What are the doubts, sir? What the doubts they have?
Prolika,
Yes,
Ashrita,
ma'am. Yes, ma'am.
Navya.
You can answer doubts.
No
No
doubts,
doubts.
ma'am.
No doubts. Oh,
Very
so
good, very good. So this is a very, 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 very interesting and also inspired, Amma.
So my classes ki they are not getting any doubts, sir.
Thank you, madam. Thank you very much.
Hey, FNDC students, why only less strength? Yeah, they are feeling he time of classes
Okay.
and they are also feeling bored.
Actually, this is a very horrible time, my dear
Yes, sir. Yes, sir.
students. But I'm very much appreciate you. So from 8 to 9 p.m. of every day. So your patience to listening my boredom class. Huh?
Not boredom, sir. Not boredom. Really, you have taken hard pains for conducting these classes and delivering the lectures to our students, sir. Not at all boredom. We are very happy to have you here on this session, sir. Thank you very much.
Thank you very much, madam, for giving me this wonderful opportunity in the faculty exchange program. So I shared my knowledge among different regions of our Telangana and twin cities of our capital city of our Telangana. So I'm very much happy to regret to give this my knowledge and also thankful to you, the management of the RBVRR Women's College Autonomous Hyderabad, Narayanaguda. And even though my college, a government degree college from Jagitial, both the principals, we are wholeheartedly thankful to them. And if any other classes also, we will plan, Madam Ji, definitely I will be back.
Yes, sir. Yes, sir. Might be in the next semester, not right now. Will be at the end of our this semester now, sir. So sure, again we'll meet you in the next semester. Okay, girls.
Okay. Shall I leave, madam?
Classes again in the next semester with new topics.
Yes, ma'am.
Yes.
Okay, ma'am. Thank you, my dear friends. Okay, may I leave?
Yes, sir. Thank you very much, sir.
Thank you. Thank you.
We look back for your sessions again, sir. Okay, girls, you can leave now.